Next, let's look at the structural differences of various lymphatic structures, starting with a lymph node. And one of the characteristic features that we're going to distinguish between a lymph node, the thymus, and the spleen is, does that structure A have a distinct cortex and a distinct medulla, and or will we find the presence of germinal centers in one or more of these lymphatic structures? Certainly a lymph node is distinct in that it has a distinct outer cortex and that is easily recognizable and distinguished from an internal medulla. And certainly the cortical region of a lymph node is characterized by the presence of multiple germinal centers, as you see in this particular micrograph. Note that in the cortex, we will find prominent numbers of lymphatic follicles or nodules. These are sites where we were going to find the germinal centers. And these are sites where B cells proliferate after helper T cells activate them by antigen presentation. The mature B cells stop dividing, migrate around the germinal centers, and contact follicular cells. The B cells with high affinity Ig migrate to medullary cords and form plasma cells that secrete antibody into lymph. The mature B cells that are not antigen specific will accumulate in the mantle area that's adjacent to the germinal center. The overall anatomy of a typical lymph node is illustrated schematically on the next slide. You can see in the outer cortical region, we find actually two zones. There's an outer cortex that is generally B cell rich, where we find the germinal centers surrounded by an adjacent mantle. The inner or paracortex tends to be the more prominent location of where we find T cells. And then finally, the medulla is a combination of medullary cords and medullary sinuses. The medullary cords, as we'll see histologically, contain macrophages, plasma cells, and lymphocytes, whereas the medullary sinuses are continuous with the efferent lymphatics that leave at the level of the hilus of the node. Note again that the flow of lymph through a node enters at the level of the cortex, and you can see a couple of efferent lymphatics entering at the level of the cortex. The entering lymph is filtered down through subcapsular and paratrabecular sinuses that are continuous with the medullary sinuses. And at various points, obviously, the entering lymph can be surveyed by lymphocytes, T and B cells, that are found in the outer and paracortical regions.